I would like to thank the organizers for giving me the opportunity to present our work on the role of the gut microbiome in hyperoxaluria. So as we all know, kidney stones are common, and the most common form of kidney stone is calcium oxalate. And oxalate is present in more than 50% of the kidney stones. And Dr. Stamatello showed in their analysis of the enhanced data that the percent prevalence of kidney stones is increasing in the United States. And also the group from Mayo Clinic showed that there is a trend in increase in incidence of kidney stones. However, they caution that over time we have been using more CT scans, which are more sensitive to detection of kidney stones, so that might confound a little bit the picture, but we think there is more kidney stones now. So as in all diseases, kidney stones are a combination of our unique characteristics. We think it's our genetic makeup and our comorbidities, and the group of exposure that we are exposed to, like our fluid intake, like our diet, body habitus, antibiotic. But there might be a new player that we recently realized might be important for kidney stone risk, which is the microbiome. It is in between a unique characteristic but can be modified with some behavior. So what is the human microbiome? So the human body is composed of 30 trillion human cells. But we also have in and on us 39 trillion microbial cells. So in reality, we are more than half microbial. At the genetic level, this is even more pronounced, where we have 20,000 human genes, but we have up to 20 million microbial genes. So we are less than 1% human at the genome level. So the microbiome is defined as the collective genome of the microbes. It includes the bacteria, the bacteriophage, fungi, protozoa, and viruses that live on and inside us. For the rest of the talk, I'm going to be focusing on the bacterial part of the microbiome, which we understand the most. So in trying to understand the diversity and functionality of the microbiome, we can know what bacteria are there, and that's usually done using 16 sRNA approaches, where we sequence a small part of the gene and we identify what bacteria are present. But we can also know what bacteria are doing, and that's through RNA sequencing, the metatranscriptome, and what protein they're producing with metaproteomics, or what metabolites they're producing through metabolomics. But we also can know what is their genetic potential, meaning what they are capable of doing. It doesn't necessarily mean that they're doing it right now, but they have the gene and the capability to do it. And that's through the DNA sequencing metagenomics. So there is multiple functions of the bacteria in the gut, but very importantly, they are known to break down food components. And one of the food component that's very important for the risk of kidney stones is oxalate, which is present in a lot of our dietary intake, and it's present in a lot of things that we think are healthy. So nuts and like all bran, spinach, and soy products. So in healthy people, we get our oxalate from endogenous production. So basically, metabolism of amino acids in the liver can produce oxalate. But we also get from our diet oxalate level that is degraded by a group of bacteria. One of them is Oxalobacter formigenes that Dr. Goldfarb spoke about earlier. But there is other bacteria like Lactobacillus, Bifidobacterium, E. coli, others that are capable of degrading oxalate. And oxalate is mainly excreted in the kidneys, where high levels can cause toxicities. So in Oxalobacter and other bacteria, oxalate enters the cell through an antiporter. It's then metabolized through the action of two different enzymes that are encoded by the genes FRC and OXY. And this metabolism produces carbon dioxide and consumes a proton that drives ATP production. So in hyperoxaluria, we can have primary hyperoxaluria, which is an increase in endogenous production of oxalate. We can have dietary hyperoxaluria if we're eating too much oxalate, or we can have enteric hyperoxaluria if we have GI pathology or gut malabsorption. But we also have idiopathic hyperoxaluria, which we don't really understand. Might be a combination of different aspects. And all of these result in higher urinary oxalate that can cause kidney stones, nephrocalcinosis, in some cases, chronic kidney disease and end-stage kidney disease. So I'm very interested in enteric hyperoxaluria because it's the intersection between kidney stones and the gut. It is a common complication of intestinal malabsorptive conditions like inflammatory bowel disease, mostly Crohn's disease. It's also common in Roux-en-Y gastric bypass, and its prevalence is thought to be high, especially in the bypass surgery patients, up to 70%. And kidney stones are common in Crohn's disease. And we think the incidence is increasing because we have more prevalence of IBD in the population and bypasses. Every year we have more bypass surgeries. So Tejan analyzed a large database, and they found that as compared to the general population, 4-year incidence of kidney stone was increased significantly, especially in Crohn's disease and more malabsorptive conditions like Roux-en-Y. And the most is in short bowel syndrome, where we have the most severe phenotype of malabsorption. So the pathophysiology, we think that if you have a lot of malabsorption of free fatty acid, that will cause chelation of the calcium, and it will allow more oxalate to be available for absorption. We think also that because of this malabsorption, or if there is inflammation of the gut, there might be increased traffic of oxalate through the gut. We also think that the microbiome, the bacteria are disturbed in this condition, and there is less capability to degrade oxalate. And there might be a role of B6 deficiency in these patients that drives oxalate production. And as we mentioned, these all can cause kidney stones commonly, but also sometimes end-stage kidney disease. So our first question was, does the microbiome affect urinary oxalate level? So our hypothesis is that the presence of the gut microbiota reduces hyperoxaluria induced by an oxalate load. And to test this hypothesis, we used mice that are either conventional, meaning they have their normal microbiome, or germ-free mice that are born without a microbiome. And we fed them 3 different diets, so normal chow, regular diet, and then 2 oxalate supplemented diet. One of them is hydroxyproline, which produces endogenous oxalate. It's a precursor of oxalate that gets metabolized in the liver. And then we followed those mice and we collected their urine. And what we found was that in this figure where we show the urine oxalate on the y-axis and the 3 different diets on the x-axis, the red is germ-free and the blue is conventional mice. We see that there is a trend for higher oxalate on all diets, but it was the most significant when the mice were fed high oxalate. And when we looked at their fecal oxalate level, that was similar, and the plasma oxalate, the serum oxalate also. So basically, in germ-free mice, you have more oxalate in the whole system. So next question was, which bacteria are oxalate degraders in the human gut? So for that, we hypothesized that oxalate metabolism is regulated by a multi-organism oxalate degrading community. We're calling it the Oxalobiome. So what we did was to look at publicly available data from healthy and IBD patients, and they had sequencing data of their microbiome, and they had metagenomic and metatranscriptomic sequencing and metabolomic analysis. And this was a large cohort of 660 healthy people and disease population, less so. So one of our talented PhD students developed this pipeline, this computational pipeline, to see what oxalate degradation enzymes are present in their microbiome. So she looked at the metatranscriptome and the metagenome to see which bacteria have the gene for oxalate degradation and which ones are actively transcribing those genes, meaning that they're trying to degrade oxalate in vivo. So first, we looked at the metagenome, and we found that there is a large number of bacteria, including Oxalobacter here, that have the oxalate degradation enzymes in their genomes, including Lactobacillus, Bifidobacterium, different species. And then when we looked at the transcriptome, meaning which ones are actively producing those enzymes, we find that the highest number was from Oxalobacter formigenes, which we know is the specialist oxalate degrader. And then we see that there are a number of bacteria that can degrade oxalate, but they're not transcribing those genes. So they're not doing this function. And then we looked at the contribution to overall oxalate metabolism at the metagenome, at the gene level, and then at the transcriptome, at the activity level, and we see that Oxalobacter seems to be the most important organism. It accounts to more than 50% of all oxalate degradation. So this was interesting. And then we asked the question, is the oxalate degrading function change in kidney stone or in hyperoxaluric patients? So there was a study by Tissinesi in 2018 where they looked at the gene OXY, the oxalate degradation gene, in the metagenome of stone formers and healthy, and they found it was significantly decreased. And they found that also this gene, when it's lower, it was associated with higher oxalate, urinary oxalate level. So that was supporting our hypothesis. We looked at 13 healthy controls, 12 calcium oxalate stone formers, 5 enteric hyperoxaluria from Roux-en-Y gastric bypass, and 7 primary hyperoxaluria patients. And we got their stool and we did analysis of their microbiome. And what we found is, in healthy people, as we showed previously, Oxalobacter seems to be doing the most oxalate degradation function. In primary hyperoxaluria, where they have a lot of excess of oxalate coming endogenously, seems like Oxalobacter is doing more function, responding to the oxalate present. But interestingly, in enteric hyperoxaluria and in stone formers, calcium oxalate stone formers, they have very low Oxalobacter level. We're analyzing the data now to see whether this correlates with urinary oxalate levels. Okay, and we also looked at the expression of those genes in IBD patients, which included ulcerative colitis, Crohn's disease, and then the phenotype of malabsorption, which is the CDL3, which has the most malabsorptive phenotype. And we saw that there is decrease in the oxalate degradation genes in all IBD patients, and that was associated with an increase in fecal oxalate levels. We didn't have urinary oxalate on this big cohort, but fecal oxalate should be a good surrogate of oxalate metabolism. So the next question was, what happens when we suppress the microbiome with antibiotic? Unfortunately, we don't have a cohort of kidney stones which received antibiotics, but we have a cohort of patients on dialysis that received, that was in a randomized controlled trial with placebo or vancomycin, and they received vancomycin weekly for 3 months, and then they were crossed over, or placebo. So we suppressed their microbiome with vancomycin. We had their blood, they don't make urine, so we don't expect that the urine oxalate would be any different. But we looked at their plasma oxalate level, and we show that in the placebo group, their plasma oxalate level was elevated, but it was stable over time. But when we looked at after their vancomycin intake, we see that all patients had an increase in plasma oxalate level. And the amount of increase at 3 months was 50%. So there was a lot of, we think there is a lot of contribution of oxalate degradation to overall oxalate metabolism, at least in humans in ESKD patients. So to summarize, I hope I've convinced you that the microbiome is important. So kidney stones are prevalent and their incidence is increasing. And kidney stones are common in patients with fat malabsorption. The gut microbiome is an important determinant of urinary oxalate level. At least we know that in animal models, we're trying to prove that in humans. The gut microbiome, including Oxalobacter and other Oxalobiome member, could reduce the risk of kidney stones if supplemented, we're hoping. And more interventional controlled human studies are needed in kidney stone forming population to confirm our observation. And I would like to thank my NYU team, my mentor, Dr. Goldfarb, and support of other collaborators and the NIH and funders. Thank you.